Hello everyone. I will be reviewing through the concepts and objectives of Unit 11, the commercialization of stem cells and stem cell products. In this unit, we will obtain a deeper understanding of the commercialization of stem cell therapies and how innovations in this field can help in many aspects of science. This unit goes over the different stem cell techniques that are used in research, treatments, and other stem cell products. To start, the FDA tightly regulates stem cell products and therapies in the United States. And there are many stem cell clinics that are located outside the US that use stem cells. Unfortunately, more of these clinics are on the rise across the United States and deliver unproven therapies. Stem cell clinics offer untested treatments that range from autism, erectile dysfunction, neurodegenerative diseases, heart failure, and musculoskeletal diseases. These clinics don't have FDA authorization for these procedures, but perform injections of adult cells that are derived from fat, skin, or bone marrow cells. Oftentimes, they obtain fat or blood cells from the patient just to inject them back, claiming that they're receiving stem cell therapy. Unregulated stem cell treatments have caused cases where patients have become paralyzed or even blind due to unwanted tumor growth from contaminated cells. Although unapproved clinics provide this service, stem cells are used to develop effective therapeutics for various diseases that currently have no cure or treatment. With this comes challenges from the commercialization of stem cells in terms of regulation, policies, and legal patents that have limited the research in this industry. Stem cells are pivotal in the future of regenerative medicine and therapeutics by allowing the ability to discover novel treatments with improving current methods. Stem cell applications have been commercialized by companies for their prominent use in diagnostics, drug discovery, and product development. Stem cells have changed the way we understand diseases and create treatments, and also how we perform medicine. Stem cells are being researched for their ability to repair damage in our bodies and help treat incurable diseases. Stem cells are used in treatments for spinal cord injuries, heart failure, and Alzheimer's. A majority of stem cell treatments are still in early experimental stages, but treatments are currently being used. Some specific types are blood stem cells for leukemia and eye stem cells for patients for eye diseases like macular degeneration. Drug development and cell therapies have multiple steps that include a discovery phase, 3 clinical trials, and a post-market phase. This process can take up to 20 years to develop a single treatment. This is because all discovery research and preclinical trials must be conducted under good laboratory practices. Drug trials must be performed under good clinical practices, and manufactured products must be formulated using good manufacturing practices. Pharmaceutical drugs are subject to new drug application approvals, and biologics undergo a biological licensing application review. The biotechnological commercial pathway is initiated with a discovery phase that includes the collaboration with academic and commercial research. This results in the necessary protection of intellectual properties with patents. After the agreement is signed, the drug or biologic is developed, tested, manufactured, and then marketed for distribution. Each stem cell therapy can cost over $1 billion within that 20 years, so this requires funding from multiple sources. A patent is the agreement to limit commercial exploitation of an invention for a period of time in exchange for publication details of that invention. Patents are required by biotech companies to attract venture capital, VC funding for their product to reach the public. Patents protect the technology behind a product, and potential investors take this patent into consideration. Strong intellectual property positions are important when trying to acquire buyers for a company. Depending on the country, there are different laws surrounding the protection of patents. Owning intellectual property is very valuable, but some issues can arise when obtaining a good IP position. In terms of a global sense, multiple countries have good protection and have wide patent landscapes. A patent landscape is the intellectual and physical property status of a technology that can be regulated within a specific country, region, or is recognized globally. Stem cell companies and institutions hold broad patents that can slow stem cell therapy development. Some researchers have stated that broad patent protections have hindered the progression of stem cell research in the last 20 years, but are now being challenged, rationalized, and harmonized to allow research moving forward. Stem cell patent regulations are different in all countries and change over time. Technology can be approved by some governments, but considered banned in other countries. In the United States, the U.S. Patent and Trademark Office, the PTO, will allow patents for purified and isolated stem cells. To be patentable by the PTO, stem cell products and research tools must meet three criteria: novelty, utility, and be not obvious. Patents have been granted for human embryonic stem cells for methods required in their isolation and maintenance. In the UK, inventing patents that involve human embryonic stem cells are more strict and placed into two categories: patents that cover the individual cells from being isolated from human embryos and the different processes that use those cells. The Patent Office grants patents for embryonic pluripotent cells because they do not have the ability to develop into a human body. For this reason, they do not grant patents for processes that obtain stem cells from human embryos or human totipotent and embryonic cells. Sometimes these approaches can impact stem cell applications by limiting the area of work that can infringe on patent landscapes. In 2001, the Wisconsin Alumni Research Foundation, or the WARF, held 3 U.S. patents on embryonic cells that included broad, significant patents on the method of deriving human embryonic stem cells and the cells themselves. They charge a fee of $100,000 and $25,000 annual mains fees to obtain a commercial research license. Geron has exclusive U.S. commercial rights for the 3 most clinically promising types of cells derived from embryonic stem cells. These include cardiac myocytes to help repair damaged hearts, neural cells to treat neurodegenerative diseases or spinal cord injuries, and pancreatic cells that could treat diabetes. Osiris has a broad patent claim protecting the use of human mesenchymal cells for any stem cell therapy. And in 2005, Advanced Cell Technologies was issued a broad patent to cover a method of nuclear transfer for non-human mammalian cloning. Although researchers were limited in their research, the WARF, the Geron, Osiris, and Advanced Cell Technology patents can only be protected for 20 years, and they soon will come to an end. A study from 2018 displays the commercialization of stem cells throughout transplantation, tissue engineering, and cell therapies. This chart shows how the $32 billion was split between different therapies. Most of the money was distributed in degenerative, musculoskeletal, orthopedic, and spinal treatments. And throughout the last 10 years, the global market for clinical research in stem cells has expanded in areas of Europe and Asia. In 2018, the majority of clinical research being conducted was in bone marrow and hematopoietic stem cells, along with mesenchymal and neuronal stem cells. Only about 1% was embryonic and 4% was adipose stem cells. Most of this clinical research is never converted into commercialized therapeutic products. One of the main reasons for the non-commercialization of stem cells can be attributed to the gap of coordination between researchers, clinicians, industry experts, and policymakers. Stem cells are being used in different methods and have various risks and rewards. Stem cells used in ancillary products or services have the lowest risk throughout the manufacturing process. Cell therapies outweigh bigger risks because they cost more and take longer to develop, but reap the benefits of discovering a novel treatment to cure patients. Stem cell treatments are currently commercialized therapies for fertility in IVF clinics and stem cell banks that hold adult, embryonic, and umbilical cord stem cells used in cancer treatments. Regenerative medicine application of stem cells have great potential, but come with many challenges. When compared to other applications of stem cells, this type of treatment is receiving patients mainly from private ventures and is high risk with low reward. In terms of stem cell therapies in humans, there are many concerns that question what type of cells should be used, stem cell characterization to maintain quality, and how many cells are required in vivo for beneficial effects. Most importantly, unregulated growth and differentiation of transplanted stem cell treatments that result in tumors or other adverse effects are a priority to prevent in patients. Some problems for the commercialization of stem cells include that therapies require rigorous and reliable protocols for in vitro maintenance, growth, and differentiation. Autologous stem cell sources help prevent graft versus host disease, but have to be maintained properly for purity and potency of cell populations. Even if your cells are not contaminated, transplanted stem cells cannot survive in large quantities forever. Commercializing stem cells in the form of services is a lower risk application, but is necessary for researchers in drug discovery, screening, and development. Stem cells can be used in pre-implantation of genetic diagnosis, stem cell banks, and optimizing drug testing techniques with humanized organoids. Stem cells have been used in creating efficient and scalable cell populations for diagnostic screening, drug safety, and toxicity predictions. along with drug target validation and other forms of tissue repair platforms. I believe it would be best for scientists to use human-derived pluripotent stem cells in drug discovery, developing, and screening. Current disease models use murine and non-human primates that do not always predict human efficacy, but using human-derived pluripotent stem cells in testing gives researchers an identical model that has similar physiological responses to humans, is genetically normalized, and can be cultured for a long time. Stem cells play a key role in personalized treatments because it allows for the ability to test drugs on humanized cells rather than directly in the patient. Researchers can use the patient's own cells to investigate how treatments react to the disease. This can be done by directly obtaining a biopsy or by collecting skin cells from the affected patient, re-engineering them into stem cells in order to conduct research, and then learning more about the disease properties outside the body. In particular, induced pluripotent stem cells are used in a disease-in-a-dish method to screen biologics for target validation, manipulation with inhibitory RNA approaches, and measuring effects of small molecule screening. Stem cells can be used to conduct ethical testing on cells for better understanding of their effects on drugs and treatments with functional organoids made from stem cells. Stem cells are used with 3D bioprinting techniques to develop living cells that can be designed from the patient's own cells into fully functioning organs. Implementing stem cells for drug development has given scientists the ability to test on human-derived cells and organoids. Stem cells provide a more accurate testing method when developing new drugs and treatments because they conduct tests on bioprinted human organoids instead of murine and non-human primate models. With the ability to 3D print stem cells into complex organoid systems, I believe the most useful implementation is testing for safety and efficacy in drug development. 3D bioprinting fabrications can be designed to mimic similar characteristics of biological, structural, and mechanical properties. Bioprinting stem cells has the potential to provide replacement options for damaged, lost, or degraded body parts. It can help relieve the organ shortage, facilitate drug testing, and make personalized medicine more advanced. Making tissues, organs, and organoids derived from patients' own cells can help increase efficacy and reduce the possibility of any side effects like graft versus host disease. 3D bioprinting stem cells are currently being used by Dr. Chen at UC San Diego through bioprinting organoids that function similarly to normal cells. These human-derived organoids are used to test drugs and diseases. With advancements in stem cell technology, scientists are able to engineer adult cells and create induced pluripotent stem cells. This overcomes ethical issues of using embryonic stem cells and can design more personalized therapies, which results in more close to the human biology. 3D bioprinting with stem cells can play a large role in drug testing instead of using outdated animal models. Human tissue, skin, and cartilage have been bioprinted, as well as organoids in the lung, kidney, and heart. These all are a great resource in surgeries and also for drug development. Along with this, stem cells were recently used to combat COVID-19 by preparing human brain organoids to test against the virus. Re-engineering adult stem cells into pluripotent stem cells and induced pluripotent stem cells can provide researchers a model for testing human-derived organoids that are ethically sourced. Human-derived pluripotent stem cells and embryonic stem cells, along with induced pluripotent stem cells, have all been utilized to make functional human cells, tissues, and organoids that are used to model disease and drug development. There's a potential approach for treating COVID-19 that is being researched that uses mesenchymal stem cells for their regenerative and immunoregulatory properties. Research has indicated that SARS-CoV-2 enters human cells via the angiotensin-converting enzyme 2 receptor, which is highly expressed in mesenchymal stem cells. Outweighing the pros and cons, mesenchymal stem cells can provide alternative treatments for COVID-19 long hauler patients. 3D bioprinting stem cells faces challenges when trying to keep cells alive during the printing process, or when vascularizing organs to oxygenate, provide nutrients, and service the cell. A common challenge faced when using organoids is the size that is limited to 1 millimeter due to nutrients being able to only diffuse through small surfaces. To develop organs, blood vessels are needed to supply nutrients to the tissues, and these designs are hard to develop. A potential way of making these new organs would be through lab-grown, fully functional human organs. Novel bioprinting techniques are being developed to combat current limitations by using organic food coloring and biomaterial, making origami organs with cellulose as the scaffolding, flash freezing 2D structures, then molding into 3D designs, using ultrasound to manipulate cell positioning, and creating bionic prosthetics with stem cells in the technology. Professor Gordon Wallace from the University of Wollongong prints customized 3D bioprinting ears with stem cells, uses a handheld 3D bioprinter to repair and replace human tissues for orthopedic surgeries, uses a similar 3D handheld bioprinting technology in optometry for ulcers, and prints skin for trauma and burn patients. Israeli scientists at Tel Aviv made a 3D bioprinted heart with the patient's own stem cells that function properly. And at Rice University, they bioprinted a heart that is designed with a vascular network that is similar and that uses natural pathways like blood, oxygen, and other vital fluids at the same small scale. Vascularizing has always been a challenge for 3D bioprinting small organs, but researchers use stereolithography with organic food dyes to shoot lights at areas of the bioink to solidify tiny designs. The commercialization of stem cell treatments is more profitable in the production of ancillary products due to cell therapy treatments taking up to 20 years and needing up to $1 billion to keep research alive. I believe that future commercialization stem cell therapies will require more coordination between pharmaceutical companies, researchers, clinicians, and policymakers. One way stem cells are being commercialized is by a company that is attempting to use them called Allergen Therapeutics. Allergen Therapeutics is a company that develops allogenic CAR T-cell therapies from iPSC-derived T-cells from a healthy donor. These cells are re-engineered and modified to limit autoimmune responses while targeting cancer cells. Allergen plans to develop allo-CAR T therapies that include Allo-501 and Allo-501A. These both target cancerous B cells by identifying CD19. Next, gene editing is implemented to knock out the T-cell receptor in order to prevent graft-versus-host disease and allogenic rejection. The CD52 gene is also knocked out to provide the engineered CAR T-cell with resistance to anti-CD52 antibody treatment. Allergen Therapeutics develops allogenic CAR T-cell therapies from iPSC-derived T-cells from healthy donors, which can help cut the cost of special techniques involved with autologous methods. The FDA granted Fast Track designation to Allo-605, a next-gen version targeting BCMA for the treatment of multiple myeloma. But more recently, the FDA placed a hold on Allergen's clinical trials because one patient that received anti-CD19 CAR T, Allo-501, formed a single chromosomal abnormality. This patient had stage 4 follicular lymphoma and was previously treated with 2 lines of immune chemotherapy and also radiation therapy. This chromosomal abnormality was reported to the FDA after they conducted a bone biopsy in order to assess low blood counts. An investigation is currently making progress to identify any evidence for clonal expansion to make gene editing the culprit for the abnormal finding. According to the reports from Allergen, the review will take a couple weeks, and the exact cause of chromosomal abnormality will be identified within months. Although there is a clinical hold, Allergen Therapeutics is not a brand new gene editing company that has failed in its first experimental trials. They have hundreds of patients that are being treated with their cellular therapies. Currently, Allergen has 5 active trials that utilize novel treatments of Allo-316, Allergen 501A, Allo-605, and Allo-715 across the United States. Allergen is also in collaboration with Notch Therapeutics to develop iPSC allo-CAR therapy treatments for treating non-Hodgkin lymphoma, leukemia, and multiple myeloma.